Hey class, in this recording we will be focusing on epithelial tissues. And as we look at epithelial tissues, these are going to be made of very closely adhering cells. Typically they'll be one or more cells thick, and we'll talk about that in a little bit more detail in subsequent slides. As we look at these tissues, it's worth emphasizing that they cover body surfaces or line the cavities within our bodies. The upper surface of the epithelial tissues are usually going to be exposed to the external environment. Or some kind of internal space. So that upper surface, referred to as the apical layer, is usually exposed to an opening as opposed to other tissue types. Our glands are included within epithelial tissues, and it's also worth emphasizing that epithelial tissues are avascular themselves. They're right next to highly vascularized connective tissues, but the epithelial tissues are avascular. And that is the underlying connective tissues with lots of blood vessels that supply the nutrients to the epithelial tissues. As we look at functions, protection is a key one for epithelial tissues. In particular, if there's multiple layers of cells, I'm thinking of a stratified squamous epithelium, that's going to help protect us from injury and infection. Our epithelial tissues are going to produce and release chemical secretions. Think of this in terms of the glandular components. Our epithelial tissues also excrete waste products. Again, another glandular component. When we look at our epithelial tissues, several different tissue types are going to have relatively high surface areas. And these surface areas are going to enhance the absorption of chemicals in the digestive tract. As we look at some of our epithelial tissues, they're very thin layers of a very thin single-celled layer, and they are very effective at filtering substances. And then finally, our epithelial tissues are going to have sensory receptors. I'm thinking of the skin in particular that has a lot of epithelial tissue in it, and we can use those to receive stimuli from our external environment. As we look at epithelial tissues, they are very close together. There's almost no extracellular matrix. They're also, because they're typically going to be exposed to some kind of a cavity or external environment, going to have a very high rate of mitosis. Epithelial tissues are going to have three characteristics present for all epithelial tissues. They include the basement membrane, the basal surface, and the apical surface. That basement membrane is going to connect the epithelial tissue to the underlying connective tissue. The basement membrane is not made of cells. Instead, it is extracellular matrix that consists of a lot of fibers. These fibers are going to include collagen, reticular proteins, glycoproteins, and other protein carbohydrate complexes. And that basement membrane will anchor the cells of the epithelium to those connective tissue cell types. Within the epithelium, if there are multiple layers, the layer that's exposed to the opening is the apical surface. The layer that is attached to the basement membrane is the basal surface. If there is only one layer of cells within the epithelial tissues, that one layer is both the apical and basal surface. Although typically we would talk about the apical side of the cell and the basal side of the cell in that single layer of cell context. As we look at the simple epithelia are going to have one layer of cells. When we think of simple epithelia, think one layer of cells. To name the kind of simple epithelia, we look at the shape of those cells. Every cell in the simple epithelia will touch the basement membrane. First we have, and then we also have stratified epithelia. Stratified epithelia has more than one layer. And as we look at these stratified epithelia, they are also going to be named by the shape of their cells, but we're going to fixate on the apical cells, the cells that are exposed directly to the cavity itself, or the opening itself. As we look at these stratified epithelia, some cells are going to be on top of each other since there's multiple layers of cells. So some of the cells are going to not be touching the basement membrane. Not all cells in a stratified epithelia touch the basement membrane. However, when we look at simple epithelia, all cells will touch the basement membrane. We can also name cells based on their shapes. So if we're looking at the cells, we can have a flattened cell type, which is called squamous, a squarish cell type, which is called cuboidal, and an elongated cell type, which is called columnar. And then when we look at these examples on the top in blue, if there's only one layer, it's called simple. If there's more than one layer, we call it stratified. And then here's the in-betweeny. This in-betweeny looks like there's more than one layer, but every single cell touches the basement membrane. So we call that pseudostratified. It looks like it's stratified, but it's not actually. It has false layers or pseudo-stratification. So we can mix and match these terms. You first will take one of the terms in red and then combine with a term in blue. And actually, I did not mean to circle columnar, so I'm just going to quick erase that with black. So I could say that I have a simple cuboidal epithelium, one layer of squarish cells. I could have a pseudostratified columnar epithelium. I could have a stratified squamous epithelium. We can mix and match the layering terminology with the shape terminology to label the kind of epithelium that we are working with. So as we look at these epithelia, we're going to focus first on simple epithelia, and then we're going to focus on stratified epithelia. The categories of simple epithelia that we're going to focus on are simple squamous, simple cuboidal, simple columnar, and pseudostratified columnar. These are going to have slides. We're going to have subsequent slides for each of these. As we look at pseudostratified columnar, it falsely appears to be stratified. I need to emphasize that it looks like it's stratified, but it is not. Every cell reaches that basement membrane, but not every cell is going to reach the free or apical surface. Within our simple epithelia, we have a lot of goblet cells. Goblet cells look kind of like, well, goblets or wine glasses. Not many people say goblet anymore. And the thing I want to emphasize about these goblet cells is they secrete mucus, and they're very common in simple columnar and pseudostratified epithelium. So let's focus on some simple squamous epithelium. The full name is simple squamous epithelium. Here's what it looks like in an unaltered photomicrograph, but this is kind of a hot mess, which is, like real life. Things are always messy in real life. So we're going to focus on just that apical surface right here, the flattened layer of cells. We're going to focus on this epithelium. We are going to ignore all of this underlying connective tissue. And I meant to go red. All of that underlying connective tissue we're going to ignore and only focus on the epithelium that I circled in green. It's a single row of thin cells, and because it's a single layer of thin cells, materials can diffuse rapidly through it or be filtered through it very quickly. These cell types are known for secreting serous or watery fluids. We have lots of simple squamous epithelium present in the alveoli of our lungs, the glomeruli of our kidneys, the endothelium of our blood vessels, and the serosa lining our body cavities. As we look at simple cuboidal epithelium, we're going to have a single layer of rounded cells. And we can see those cells highlighted over here. The single layer of rounded cells can function for both absorption and secretion. We have some mucus production, and in particular within our kidneys, we also use them for movement. We have cilia that are coming off of them that can help to move substances past them. We find that we have simple cuboidal epithelium present in our livers, thyroid glands, mammary and salivary glands, the bronchioles of our lungs, and the tubes in our kidneys, the renal tubules. As we look at our simple columnar epithelium, we have a single layer of tall cells. So here's one, simple meaning single, columnar meaning long. And this particular simple columnar epithelium is also going to have microvilli present, giving it a brush border appearance. As we look at the simple columnar epithelium, it also has a goblet cell that has the wine glass appearance. And these are beautiful looking goblet cells. So here's the, kind of the wine glass. There's one wine glass. Here's another wine glass for our goblet cells. And I know I'm really bad at drawing wine glasses. As we look at simple columnar or columnar epithelium, the nuclei of a columnar cell is typically going to have an oval appearance. They can work for both absorption and secretion. And we find that if there are those goblet cells present, they also are going to have secretion of mucus. We find them lining the gastrointestinal tract, the digestive tract for short. They are present in the uterus, our kidneys, and the uterine tubes. You may know the uterine tubes as the fallopian tubes. We also have a pseudostratified columnar epithelium or pseudostratified epithelium. So as we look at this pseudostratified epithelium, it's going to have some goblet cells present. Let me switch here so I have my pen. We have our goblet cell. We're also going to see that we have some more cilia, particularly on this membrane. And the cilia are longer than the microvilli. And this pseudostratified epithelium looks multi-layered, but every cell is going to touch that underlying basement membrane, which is right here. I'm tracing the basement membrane in green right now. We find that with the pseudostratified epithelium, we have nuclei present at multiple heights, and it's going to secrete mucus and propel the mucus. The goblet cells will secrete the mucus, and the cilia will move the mucus. We have pseudostratified epithelium present within our respiratory tract. Think of the lining of our respiratory tract. And some portions of the male urethra will also have pseudostratified epithelium. As we look at stratified epithelium, they can have 2 to 20 or even more than 20 layers of cells. These cells are going to rest directly on top of other cells, and only that deepest layer will be attached to the basement membrane. Most of the cells within stratified epithelia do not connect to the basement membrane. We have 3 categories of stratified epithelia: stratified squamous, stratified cuboidal, and stratified columnar. And they're named for the shape of the cell that's exposed on the apical surface. That's the key layer of cells that determines the second word within the name. It's worth emphasizing that stratified columnar is very rare. I personally have never seen stratified columnar cells on a microscope slide with my naked eye. I've seen pictures of them online, but finding them is very difficult. We also have an in-betweeny-weeny called transitional epithelium that I'm circling in red. This is a hard to categorize one because the cells change shape and the number of layers also is altered depending on whether or not that tissue type is stretched out. As we look at these tissue types, stratified squamous is very, very common. It's the single most common epithelial tissue in our body. We find that the bottom layer, that basal layer, the deepest layer, is going to have cells continually reproducing and dividing, undergoing mitosis. The daughter cells produced are going to be pushing upwards towards the apical surface, and as they move towards that apical surface, they increasingly flatten out and assume the squamous appearance. Ultimately, they die and flake off. If they flake off, sometimes we refer to that as exfoliation. The technical term for exfoliation is desquamation. And as we look at these stratified squamous, there's 2 kinds. There's the kind on the surface of our skin that's filled with keratin. This is a keratinized stratified squamous epithelium. And because of that keratin, it's very exceedingly abrasion resistant. Non-keratinized stratified squamous epithelium lacks that layer of dead cells filled with keratin. But because it's stratified, it still does have resistance to abrasion, just not as much resistance to abrasion. Here in this figure, we can see keratinized stratified squamous epithelium. This is thick skin from the bottom of our foot. And that we start down here, and we end over here. And I would venture that, oh my Atlanta, I would guesstimate there's at least 150 layers of cells from from one end to the other end of these layers. And as we're looking at this, those cells at the bottom, at the basement membrane, are rather round, but as they work their way towards the apical surface, they become flatter and flatter and flatter. We find that we have keratinized stratified squamous epithelium present on the surface of our bodies. In particular, on our palms and the bottom of our feet, we have thick skin that has excessive keratinization. And that's good because the palms of our hand and the bottom of our feet are exposed to an excessive amount of abrasion relative to other parts of our bodies. We also have non-keratinized stratified squamous epithelium. This tissue type is the same as keratinized, except it doesn't have that layer of cells on the surface filled with keratin. So it's lacking that surface layer of dead keratinized cells. It can resist abrasion and penetration of pathogens because there's multiple layers of cells protecting the layer or the membrane, and there's always new cells pushing their way up from the basement layer up to the apical layer. We find that we have non-keratinized stratified squamous epithelium in our oral cavity, so in our mouth, on our tongue, within our esophagus, and lining the vaginal canal as well. We also are going to have stratified cuboidal epithelium. Stratified cuboidal epithelium is multiple layers of roundish cells, and these stratified cuboidal epithelial cells are going to be typically associated with glandular secretion. So we have secreting sweat, producing sperm, producing ovarian hormones. They're going to be producing something that leaves the cells. And the secretions will go into an empty space referred to as the lumen. I just circled the lumen right here in green. So here's one lumen, here's a second lumen, there's a third, and there's a fourth lumen. And we find that these cells are going to be primarily associated with glandular secretions. I think I've said that 3 times now. We also are going to have transitional epithelium. Transitional epithelium is characteristically going to be associated with cell tissue types that stretch out a lot. So we have a multi-layered epithelium. Here is our band of transitional epithelium. And this transitional epithelium is a multi-layered surface or multi-layered epithelium. And the cells go from rather roundish when they are when the tissue is non-distended, to being more elongated as that tissue stretches out. And also something else that occurs as the tissue stretches out is that it looks like there is a reduction in the number of layers of cells. So this allows for us to fill the urinary bladder and also to have our ureter stretch out mildly as well. That's all we have for this recording. If you have any questions or comments, please feel free to post them on the class discussion board or to shoot me an email. And as always, happy studies.